Hi there, I'm Ruan Parrott and I'm currently working on my PhD with the University of Cape Town, South Africa. I'm excited to show you the work we've recently started delving into. Everything I'll be showing you in this presentation is based around the Southern Ocean seasonal experiment, named the Scale Cruises, that took place over austral winter and spring of 2019. So today I'll focus on one component of the data we collected, specifically the high-resolution surface phytoplankton community composition and the implications for biogeochemical cycling across the Atlantic Southern Ocean, with a specific focus on the phytoplankton group-specific contributions to chlorophyll-a and particulate organic carbon in the surface ocean. As you can see, the specific dataset alone involved a multinational, multi-institutional collaboration. The unique advantage we have as South African researchers lies within our access to the advanced icebreaker research vessel, the SA Agulhas II, shown here in these images. With the SA Agulhas II, we depart from Cape Town along the Good Hope line, pictured here on the left, and cross the southern Atlantic sector of the Southern Ocean towards Antarctica. This region is characterized by very distinct physical zones. The gradients of change between these zones are defined by the fronts shown here. As we travel southwards, we cross the subtropical front, subantarctic front, the polar front, and the southern Antarctic circumpolar current front. However, during winter and spring, we encounter the Marginal Ison, that's shown here on the right. This is the seasonal expanse of frozen surface sea ice that forms during winter, extending right up past 60 degrees south, then melting again towards austral summer. Our knowledge of the region's biogeochemical properties and cycles are limited, specifically with regards to the effects of seasonality. This is owed to the fact that the whole region is highly inaccessible, especially during winter. So, as you can see here, during the two separate winter and spring scale cruises, we got as far south as roughly 55 degrees south, where we encountered the Marginal Ison. The data I'll be showing here will be a rudimental comparison of the southward journeys of these two cruises. The full extent of the scale cruises, however, were composed of several months' worth of sea time and encompassed a vast array of experiments with multiple teams from all over the world, all focused on studying the Southern Ocean region and the Marginal Ison. The objectives of this study is to gain an understanding of the phytoplankton community composition between seasons at an individual cell level, including details on cellular carbon and chlorophyll-a content. Additionally, we'd like to assess the implications of these changes in community composition for carbon export and nutrient cycling in the region across winter and spring. This kind of data is useful in training climate change prediction models and ground truthing satellite measurements for the region. As I mentioned before, I'm just going to show our preliminary new data as an introduction to our data-rich compilation collected on the southward legs of our cruises. At 2-hour intervals on both cruises, we took underway samples, surface water samples, for the following analyses: flow cytometry, chlorophyll-a concentrations, nutrient concentrations, which include nitrate, nitrite, ammonium, silicate, and phosphate, and natural abundance isotopes of nitrate and ammonium. At 4-hour intervals, we collected natural abundance isotopes of particulate organic nitrogen and carbon. Twice daily, we performed uptake experiments with the following nutrients: nitrate, ammonium, urea, and inorganic carbon. Additionally, a thermosalinograph provided a continuous dataset of temperature and salinity to provide the physical data for our work. It must be repeated here that there is a heap of work not mentioned, including but not limited to trace metal and zooplankton measurements and experiments. To characterize the phytoplankton community, a cytobuoy pulse shape recording flow cytometer was used. Cells are pumped through the flow cell to pass through the laser, shown in the image on the left, one at a time. Each cell gives measurements of their exterior and interior characteristics to determine what they are and what they are composed of, whilst images of selected cells are taken. On the right-hand side, you can see images of a dinoflagellate and nanoflagellate, each with their accompanying pulse shapes. Our main focus here is on the forward scatter, or FWS, and red fluorescence, FL red HS signals we record from every single cell. Forward scatter intensity is proportional to cell volume, and its pulse shape reflects the cell morphology. This is used to determine the carbon content of each cell. Red fluorescence is proportional to cellular chlorophyll-a concentrations and can be used to estimate the potential productivity of individual cells. Looking at some of our results now, we can see at a glance that our temperature and silicate concentrations did not show much change from winter to early spring. We see the expected drop in surface water temperature as we travel southwards, coinciding with an increase in silicate, especially at around 48 degrees south. Similarly, for nitrate, there seems to be no noticeable difference in concentrations between winter and spring. Surface nitrate concentrations increase almost linearly with the journey southwards. Though nitrate and silicate don't seem to show much difference between winter and early spring, ammonium concentrations do. If you look at the differences in the y-axis, winter ammonium concentrations are almost double, and more so than that south of 48 degrees south, than those measured in early spring. This is consistent with winter ammonium accumulation. This follows light inhibition and heterotrophic activity during winter, and the onset of spring productivity leads to lowering of ammonium concentrations in the surface ocean as light likely becomes less limiting. This becomes very evident when we look at the chlorophyll-a concentrations. In winter, chlorophyll-a decreases steadily with the journey southwards. However, in spring, probably with an increased availability of light, chlorophyll-a concentrations are consistently higher throughout the transect south of 37.5 degrees south. At the most southern edge of the transect, at the ice edge, chlorophyll-a concentrations are very noticeably higher. This is a likely indicator of the spring bloom onset as surface sea ice starts to melt. Using the flow cytometry data, we can get a detailed look at what phytoplankton groups are present and in what quantities. The groups identified within our data are as follows: coccolithophores, cryptophytes, diatoms, dinoflagellates, heterotrophs, nanoflagellates smaller than 5 micrometers in diameter, nanoflagellates between 5 and 8 micrometers, nanoflagellates between 8 and 15 micrometers, other microorganisms larger than 15 micrometers, picoeukaryotes, and picoprokaryotes. Total cell abundance in cells per mole are on the left, relative cell abundance on the right, with winter populations on the top and spring on the bottom. The x-axes of these plots are arranged in order of sampling, so from Cape Town down to the Marginal Ison. There is a trend towards decreasing total abundance from north to south, and it's evident that phytoplankton dominate in total abundance across both seasons, although nanoflagellates smaller than 5 micrometers in diameter become more dominant south of the polar front, especially in winter, showing a tendency for larger cells to dominate in this region. Total cell abundance roughly doubles from winter to spring, with whilst picoplankton expand their geographical range southwards. Diatoms clearly become more abundant south of the southern ACC front, or the Antarctic Circumpolar Front, coinciding with a spike in chlorophyll-a concentrations in this region. Using the relationship between measured forward scatter and cellular volume, carbon biomass per cell was estimated using the generic protist formula from Mendendorfer and Lezard 2000. Looking at the total biomass plots on the left, we can see that they are on the same scale, illustrating the striking difference between winter and spring measured biomass. Most notably, the diatoms that appear in early spring south of the southern Antarctic Circumpolar Front create a massive amount of carbon biomass, showing potential for large carbon export rates, with the onset of the spring bloom. Let's fix the scale so we can, on this first plot, so we can see more detail. It becomes clear that there is a general trend towards lower biomass as we travel southward in winter. But this does not seem to be the case in early spring, as diatoms become much more dominant across the entire transect, increasing the total biomass across the system. Interestingly, heterotrophic microorganisms had a larger contribution to total biomass north of the subantarctic front during winter. This, however, shifted with the dinoflagellates and small nanoflagellate populations to the open Antarctic/subantarctic zone in spring, where diatoms were found to be less dominant. As mentioned before, red fluorescence is proportional to cellular chlorophyll-a concentrations and can be used to estimate potential productivity of individual cells. It must be noted here that the total red fluorescence of the winter cruise plots is on a different y-scale, or y-axis scale, to the spring cruise plots. Total red fluorescence in winter was less than half that found in spring. Red fluorescence followed the same general decreasing trend toward southwards in winter as abundance in biomass did, illustrating the dominance of smaller cells during winter. Nanoflagellates smaller than 5 micrometers also increased in dominance southwards during winter, and just as seen in the biomass, diatoms became the dominant group south of the southern ACC front. Although it must be noted that the smaller cells played a larger role in the relative contribution of red fluorescence than they did in the biomass, illustrating their higher chlorophyll-a content per cell and their importance in total productivity. Indeed, low biomass doesn't necessarily imply low productivity within each cell, and this concept is very important in understanding the complicated dynamics of nutrient cycling and carbon export. This is also very evident in the open Antarctic/subantarctic zone in spring, where phytoplankton dominate abundance, fell away in biomass due to their small sizes, and then seem to dominate in red fluorescence within the region, making it as productive as surrounding regions that exhibit slightly higher biomasses. Note that the y-axis is different for each of these plots. In fact, biomass increases for each of the groups shown below between winter and early spring. When we look at the individual groups and their distribution along the transect, we can pry out some interesting observations. For example, coccolithophores, whose distribution is said to show the great calcite belt surrounding Antarctica, are seen to move southward in early spring, whereas diatoms, considered to be the main drivers of the ocean's carbon export pump, due to their heavy silicate structures and preference for nitrate uptake, are somewhat evenly distributed in winter, but struggle to grow south of the southern ACC front. Their distribution in spring turns to being averted from the open ocean, preferring to grow next to the Marginal Ison and close to the South African coastline. And finally, dinoflagellates, a group that can be heterotrophic, autotrophic, or mixotrophic, seem to remain open water specialists, building most of their biomass in the subantarctic zone that lies between the subtropical front and the subantarctic front, even as it shifts between seasons. Moving forward with this work, we will further evaluate the relationships between community composition, carbon biomass, and chlorophyll-a concentrations with regards to carbon export potential and nutrient cycling within the surface ocean of the Atlantic sector of the Southern Ocean to provide data for the training of predictive models, ground truthing of satellite measurements, and for any future work in the field. Thank you very much.